How do you stay safe?
It's so not dangerous. The world is not a dangerous place. You can go to Washington, D.C., and I'll show you a street where you should not walk down it, even in the middle of the day, right? So in every major city, there's like streets where there's people shopping and it's nice, and then there's places where it's just, if you have common sense, the world is not a dangerous place. It's not like the media makes it out, or the movie Hostel, where you're going to get vivisected. Yeah, it's pretty, okay. But now for something completely different. Let's talk about the mechanisms of microbial genetics. Yeah. So we're basically just going to unpack I think it's Francis Crick's central dogma that said that the blueprint, the software, is encoded in DNA, and that is changed or transcribed into RNA, and that is what binds to ribosomes to be translated into protein. Life is protein. Most of this stuff is protein, and what isn't was made by enzymes, which of course from the previous chapter we know are a type of protein. So let's get to it. We're going to focus on DNA, what it looks like and how it works, gene expression, and a little bit about what happens if you tweak it, mutation and stuff like that. Okay, so again, central dogma, DNA, across encryption, across code. We're going from a language written in A, G, C, and T nucleotides to a language written in A, G, C, and U nucleotides. But it's the same message, it's just in a different language. Transcribed, transcription. And that will actually be flushed out to its meaning, the protein. That will be translated. So we're just going to look at these steps in detail. A little more detail than we covered in Chapter 10. And again, this can be summarized as gene expression. So all humans have the same genes. Any two human beings are about over 99% identical. So why don't we look identical if we have the same blueprints? It's like having the same library with the same how-to manuals in it, but different individuals pull different how-to manuals off of the shelves. We all have the how to make blue eyes, but not all of us actually express that gene. Okay. It's a little more complicated than this without getting into epigenetics, where it's kind of like Darwin said, no, he didn't know about genes, but he said there are these units of inheritance that are passed on. He didn't know it was genes in the form of DNA, but he knew something was, the information was being passed on. Lamarck said, no, the more you use or disuse body parts in the next generation, they'll be enhanced, or the opposite of enhanced, dehanced. Like, the reason giraffes have long necks is not because the parents passed on genes that told their necks to grow bigger, but because they were stretching their necks out every generation. So if I work out and get really muscular, my children will be more muscular. That was found to be poppycock, but now with this new field of epigenetics, it's actually, there's a little bit of truth to it, just a little bit though, and it makes it very confusing. We won't get into it in 100 level class. But so you're not a product of your genes nor a product of your environment, nature or nurture, kind of both. So the chapter talks a tiny bit about epigenetics. And you might have genes to make pigmentation, but if you never go out in the sun, you're not going to have, tan skin, for example. So it's not only what genes you have, but it's under which environments the genes are expressed. Just because you have certain genes doesn't mean you're going to express them. As we'll get into later, in simple models in bacteria, we'll really flush this out when we talk about the operon theory of how genes are regulated. But remember, in the last chapter, we said if you took apart one of your chromosomes and stretched it out, remember, a chromosome is just a bunch of coiled up DNA around proteins, histones, and whatnot, like thread around a wooden spool. If you took all that out, about 98% of it doesn't contain areas that are ever transcribed and translated. So there are these huge areas they used to think were junk. Now we're realizing so these genes tell you what to make. These areas tell you when to make them. So environment plays a big role in determining when to make things. Okay, so before we get into the central dogma of transcription translation, let's talk a little bit more about replication. What is replication? It's often summarized as DNA. Remember, these arrows they don't mean turns into. A blueprint doesn't turn into a house. It's used to make. So DNA being used to make more DNA. Basically, the double helix is going to open up, and each strand is going to act as a template to build the other strand. Because remember, A only matches with T, and G only matches with C. So if you know one strand, you're guaranteed to know the complement of the other strand. That's about as far as we took it last time. Let's talk a little bit about the subtle nuances of replication. It is thought to be semi-conservative. What does that mean? It means each old strand of the parent is used to make, as a blueprint, to make the new strand. So each new strand of DNA is half old, half new, half old. It's not a mix. There's all these other theories. It was found out. Again, there's two basic principles to investigating the molecular world. Well, to investigating a lot of stuff. One, break things and see what happens. To find out the function of said things that you broke. Break them and compare them to a non-broken thing. You can smash the headlight and drive the car around as opposed to a regular car, and you'll realize, oh, they can't put out light when it's dark. So you find out, oh, headlights produce light. Another way of doing it is radio labeling things and seeing where they go. So by radio labeling the parent strands, they were able to find that the whole thing was conserved. Each daughter strand was partially radioactive, showing semi-conservative replication. Okay, so you need to know the enzyme that copies DNA. It's called DNA polymerase. What do you think the enzyme is that makes RNA? The enzyme that makes DNA polymerase. The enzyme that makes RNA? RNA polymerase, right? It's not terribly tricky. Now, there are several types of DNA polymerase. I'm surprised we went into this. This book wasn't really necessarily written for a 100-level course, so I'm kind of trimming the stuff that you'd cover in 300 level and higher. There are three types, or there's more than three types of DNA polymerase. It's really polymerase number 3 that does replication. 1 and 2 were used to repair damaged segments. I don't know how important that is, really. But here's really what you need to know about replication. You need to know, besides replication being semi-conservative, DNA strands run in opposite directions. So remember, you can only read these letters, R-E-P-L-I-C, to make sense going that way. If you read this N-O-I-T-A-C, it doesn't make any sense. Same thing with the DNA strands of A, G, C, T, T, G, A, C. You can only read them from the phosphate end towards the sugar end, or the 5'. Well, you read it from 3. It's built from 5 to 3. Let's focus on that. I think I have a picture here. Do I have a picture of a DNA nucleotide? Doesn't look like I do. You might remember, hopefully, from the last chapter, that DNA, each nucleotide of DNA, the A, G, C, and T, it has a phosphate group and then a 5-carbon sugar that's either deoxygenated in DNA or a ribose RNA. And then it has a base, either a purine or pyrimidine that can, that's the part that's called A or G or C or T or whatever. So the carbons are labeled 1, 2, 3, 4, 5. So the carbon closest to the phosphate is the 5' carbon. The carbon closest to the sugar end is the 3' carbon. And the two strands of DNA, when you unwind the double helix, one of them is going to be located like this: phosphate, sugar, A, G, C, T, 5' end, 3' end. The other one is going to be running upside down, or anti-parallel. So this would be the phosphate end, and this would be the 3', the sugar end. So this A binds to that T upside down. This G binds to that C upside down. Why is this important? This may unlock the secret to immortality. This simple concept. Now, just as you can only make sense of this reading from left to right, the AGCT software can only be made sense of reading it in one direction. So, to replicate, first there's an enzyme that unwinds the DNA. I thought we talked about all this stuff in one of these slides. The names of the enzymes now. Well, gyrase unwinds the DNA, helicase. I swear I put that stuff in here. Oh, here we go. So yeah, these should also be pretty important words. Okay, I saw a bumper sticker one time that said, "Girl, I wish I was DNA gyrase so I could unzip your genes," or something. I thought that was pretty clever. So remember, it's all twisted up. You got to unwind it, and then they call it like a ladder. And then you got to break the hydrogen bonds between the A, G, C, and T to make a replication fork. So it's unwound, and then it kind of opens up. Let's draw it a little differently. Where it's double-stranded here still. A is matching with T's, G's with C's. But then it opens up, and it's single-stranded. Remember, we said this one was running phosphate to sugar. This one was running phosphate to sugar in the opposite direction. Replication, the enzyme that replicates DNA polymerase, it will only add nucleotides from 5 towards 3. All of these processes, replication, transcription, translation, always go from phosphate towards sugar. That's the way they make sense of the molecule. You can't say left and right because this is happening in a cell where there is no left-right, right? So anyways, the problem here is this strand, no problem. So if this is an A, and this is a T, and a G, and a C, and a C, and an A, DNA polymerase can bind and start adding from the 5' end, tick, 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 towards the 3' end. Right? It'll grab a T floating around, it'll grab an A, it'll grab a C, it'll grab a G, a C, or a G, and a T. Boom, right? At about, in prokaryotic cells, about 1,000 a second, matching up G's to A's and T's to C's, G's to C's and T's to A's. In our cells, in eukaryotic, it's only about 100 nucleotides a second. It's about 10 times slower. But that's called the